Hi. In this particular video, we are going to discuss and compare the mechanisms of action for peptide and steroid hormones. We will have some comparative features as to how those hormones are synthesized, secreted, what are their half-lives and so on. We will compare the features of these two categories of hormones. Before we start doing that, let's just quickly recap as to what are the types of hormones. Hormones basically fall into three categories. One category of hormones is called as peptide or protein hormones, which will have the chains of amino acids. So there would be long chains. For instance, growth hormone is 191 amino acids. So all these hormones are peptide or protein hormones: growth hormone, insulin, ACTH, TSH, FSH, LH, glucagon, PTH, all those. Second category is steroid hormones. Steroid hormones have a CPPP ring, cyclopentanoperhydrophenanthin ring, and they are synthesized from cholesterol. The third category is derivatives of a single amino acid, and that single amino acid is tyrosine. So the hormones that are derived from a single amino acid tyrosine are thyroid hormone and catecholamines, dopamine, noradrenaline, adrenaline. So that is basically the categorization for all the hormones. Let us describe and compare the peptide versus steroid hormones. First thing, before we start actually the comparison, one must understand that the peptide or protein hormones such as insulin, growth hormone and the other ones, PTH, glucagon, etc., they are not lipid soluble. They are water soluble, but they are not lipid soluble. So the point is that they cannot cross the cell membranes easily. You know, our cell membranes are made up of lipid bilayers, but since these are not lipid soluble, they can't cross the cell membranes easily. This should be kept in mind. Compared to that, the steroid hormones, testosterone, estrogen, progesterone, the glucocorticoids, basically steroid hormones are synthesized from specialized cell types. They are synthesized in this specialized glands such as adrenal cortex gland, which has specialized cell types in the sense that they have the enzymes specifically meant for synthesis of steroid hormones. So adrenal cortex, we will have mineralocorticoids such as aldosterone, glucocorticoids, the cortisol, and the sex steroids. So we'll have testosterone, estrogen, progesterone, which are synthesized from the reproductive organs as well, testes and ovaries. Now, steroid hormones are lipid soluble, which means they can cross the cell membranes easily. Cell membranes which are made up of lipid bilayer. So that is a basic fundamental difference which should be understood so that we can understand this comparison. Let's start and understand this comparison. First point is that the peptide and protein hormones, they are synthesized and stored. They are synthesized and stored in those particular glands, and only when a stimulus arrives that they are secreted. This is called as stimulus secretion coupling. Why it happens like this? It's because they are stored inside these cells which have synthesized them, and since they cannot cross the lipid bilayer and come out of the cell easily, they can be stored. Compared to that, what about the steroid hormones? Steroid hormones are synthesized and then not stored. They are immediately released into the circulation. Synthesized and immediately released into the bloodstream because, as we've already seen now, that they are lipid soluble, so they can cross the cell membrane easily, they can come out, so they can't be trapped inside the cell for long. They will immediately come into the circulation. That's the first fundamental difference between the two. Well, the point to be noted here is that the steroid hormones, since they can't be stored and later on released, the regulation is about their synthesis. This is a very important difference. In the case of peptide or protein hormones, their secretion is regulated. The stimulus will arrive and then they will be secreted. So, secretion has been regulated, whereas in the case of steroid hormones, their synthesis is regulated. That is, for instance, the enzymes necessary for the synthesis of steroid hormones will be upregulated or downregulated, and in this manner, the synthesis will be regulated in the case of steroid hormones, whereas secretion will be regulated in the case of peptide or protein hormones. A very important difference. Second difference is a minor one actually, but still needs to be understood, as peptide or protein hormones are synthesized as prohormones or precursor hormones, whereas steroid hormones, generally speaking, are synthesized as active hormones. Although there are exceptions to that as well, but by and large, if you see, by and large, this can be said to be another differentiating feature. Third is, how do they circulate in the plasma? The peptide or protein hormones, they circulate as free hormones, unbound to plasma proteins. It's quite obvious. I mean, these are protein hormones, so they are not bound further to proteins in the plasma. They remain free in the plasma. Whereas the steroid hormones, they are bound to proteins in the plasma. For example, there is CBG, cortisol binding globulin, or such proteins which bind those steroid hormones. And as a fallout of this, the next comparative feature is that the peptide or protein hormones, they will have short half-lives in the plasma. Growth hormone, short half-life in the plasma, about 2 minutes or so. Insulin, about 3 to 5 minutes or so. So, they have very short half-lives in the plasma. Compared to that, the steroids will have longer half-lives because primarily that they are bound to proteins in the plasma. Now, here I would like to point out an exception. Protein hormones are not bound to plasma proteins. They are free. There is an exception to this, and that exception is IGFs. The exception is insulin-like growth factors. Insulin-like growth factors, also called as somatomedins, they are the mediators of growth hormone action. They are synthesized by the liver, and these, although they are proteins themselves, they get bound to plasma proteins. They get bound to the proteins in the plasma, which means what? Which means they have longer half-lives. There are 6 types of IGF-BPs discovered so far, IGF binding proteins. So IGF-BPs, 6 variants have been discovered, and because of these binding proteins, these IGFs, insulin-like growth factors or somatomedins, have longer half-life. So just we can understand now that growth hormone itself has got a short half-life, but what it does in that short half-life is that, or short life in the plasma, is that it goes to the liver and it causes release of IGFs, and then the IGFs, having longer life in the circulation, they will exert the effect of growth hormone because they have got longer lifespan in the circulation. Now that's another comparing feature between the two. Next, another important characteristic that separates the two is about the receptor location. Having already said that peptide or protein hormones cannot enter the cells easily, the target cell, they can't enter the target cell easily. Therefore, their receptors are located in the cell membrane itself. The target cell has to be acted upon by the peptide hormone, but it cannot enter the cell because it is lipid insoluble. Therefore, its receptor is located in the membrane itself. And when hormone binds with the receptor in the cell membrane, there is activation of enzymatic machinery inside the cell, such as adenylyl cyclase enzyme will be activated and it will convert ATP into cyclic AMP, just for the example. And then cyclic AMP, for instance, or cyclic GMP will act as second messengers. So if the hormone is the first messenger, then there is activation of second messenger inside the cell. That is the action of protein hormones, how they can exert without entering the cell, is by acting on the membrane receptor. So that's for the protein hormones. So far as the steroid hormones are concerned, their receptor is, or their receptors are intracellular receptors, because the steroids can cross the membrane easily. They cross the lipid bilayer, they enter the target cell, and the receptor is inside the cell. Now, where inside the cell? Is it cytosol? Is it cytoplasm, or is it the nucleus? Some of them have cytoplasmic receptors, some of them have nuclear receptors. So, for instance, estrogen is known to have a nuclear receptor, intracellular receptor meaning nuclear receptor. But it has been observed that the hormones like estrogen, which have intranuclear receptor, they too have a cytoplasmic receptor, intracytoplasmic receptor, or it has been called as primary receptor. So what happens is estrogen will enter the cell, target cell, combine with this primary receptor, and then hormone receptor complex will enter the nucleus. Or in some of the other, or even in the case of estrogen, they have found that it's not necessary that it binds with this receptor. It's not always that it has to bind with the receptor. Intranuclear receptor means estrogen will straight away enter the cell, travel through the cytoplasm, enter the nucleus, and then it will bind to the specific DNA strands. Or if it has combined with that primary receptor, then the hormone receptor complex will enter the nucleus, and they will bind to the so-called hormone response elements inside the nucleus. So attaching to the specific strands of the DNA, and that's how they will cause the protein synthesis. They will induce the DNA transcription, and protein synthesis will be, new proteins will be synthesized. So that's how these steroid hormones, they exert their action via the receptors. Why I mentioned about the estrogen or the other steroid hormones? there is a fair amount of debate whether cytoplasm has any receptor for them or they straight away enter the nucleus. There is a growing evidence that the steroid hormones, many of them, they do have even the membrane receptors, cell membrane receptors. There is evidence to that effect. This is recent advances, and therefore they do combine with those receptors as well. The research is going on this, but estrogen receptor is an for the sake of formality, we can say that estrogen receptor is a nuclear, intranuclear receptor, because finally it's going to actually bind to those hormone response elements inside the nucleus. Mineralocorticoids, aldosterone has the receptor called as mineralocorticoid receptor. Estrogen receptor inside the nucleus would be ER or MR. These are the intranuclear receptors for those steroid hormones. So that is a comparative analysis of the peptide versus steroid hormones. One last point of comparison is actually not a comparison, but an additional feature which we can say about each of these categories. For the steroids, there is something called as post-release modification. That's another feature seen in the case of steroid hormones, that there is post-release modification. Means once the hormone has been synthesized and it has been released, it can get modified further. Let me give you two examples. In the first instance, in the first example, when the hormone gets further modified after its release, it can form another hormone of the same class, of the same category. Example, vitamin D. Liver forms 25-hydroxycholecalciferol. It's released. It's a hormone. Now it will go to the kidney and it will one hydroxyl group further will be attached. One more hydroxyl group will be attached so that it forms 1,25-dihydroxycholecalciferol. So post-release modification, forming another hormone of the same type, same class. That's an example. So 25-hydroxycholecalciferol as well as 1,25-dihydroxycholecalciferol, they both have the hormonal actions. The other example of post-release modification is the peripheral conversion of the steroid hormone to form another hormone which is of a different category, of a different class. For instance, testosterone can be converted peripherally into estrogen. So this is an example of peripheral conversion of the hormone after it's released into another hormone of another class, a different class or a different category. That's another important feature of steroid hormones. So far as the peptides are concerned, is there any parallel to this? Any parallel feature? Let's see. In the case of protein or peptide hormones, they are synthesized as precursor hormones, pro-hormones or pre-pro hormones, the precursor hormones which are inactive hormones. So pre-pro hormone is synthesized and then there will be a proteolytic cleavage to form a pro hormone, and then finally hormone will be synthesized. Rather, finally hormone will be released. Synthesis was that of inactive hormone precursor. In this, we find different types of examples. For instance, from the precursor hormone, just one copy of the hormone is released. Example, insulin. Insulin synthesized by the pancreatic beta cell as pre-proinsulin. Now there is a proteolytic cleavage to form proinsulin, and then proinsulin will be further split to form insulin and connecting peptide or C-peptide, and they will be then released into the circulation in equimolar concentrations. In fact, the levels of C-peptide in the circulation are sometimes taken to be the level of beta cell activity of the pancreas. Now, this is one example, but just one molecule of that hormone or one copy of the hormone was formed from the precursor hormone. Second example, from the precursor protein, multiple types of hormones can be formed after the cleavage of the precursor hormone. Example, POMC, pro-opiomelanocortin. This pro-opiomelanocortin, synthesized by the intermediate lobe of the pituitary, there are basophils in there, they will synthesize this pro-opiomelanocortin. Now, its first proteolytic cleavage will form corticotropin or ACTH and lipotropic hormone, and then the further cleavage will form MSH, CLIP, endorphin. So, multiple hormones or multiple types of hormones form from one single pre-pro hormones, one single precursor. This is the example. By the way, just note this point that ACTH and MSH, they share their first 13 or so amino acids, and therefore ACTH has MSH like activity. In the Addison's disease, when by negative feedback ACTH levels increase, there is pigmentation. Why? Because ACTH has this pigmenting activity of MSH. They are derived from the same precursor, they share the sequence homology, or they have sequence homology, and therefore that's the reason why there is pigmentation when ACTH levels increase. However, that's not the point for discussion. The point was that one precursor and multiple hormones. There is one more variety that can be seen in the case of protein hormones. One precursor forms multiple copies of the same hormone. Example, TRH, thyrotropin releasing hormone. It's synthesized as pro-TRH as a large polypeptide precursor, and then from its proteolytic cleavage, 6 copies of TRH will be synthesized from this. So, multiple copies of the same hormone. Well, by the way, these precursors are therefore sometimes called as polyproteins. Which are forming multiple copies of the same hormone, or multiple types of hormones derived from a single precursor, or one copy of that hormone from one precursor. That is about the protein or peptide hormones, and this is the comparative analysis of peptide versus steroid hormones.